Okay, so in the earlier lecture, we talked about the overview. In the overview, we talked about the two types of the immune response, the adaptive response and the innate immunity. So in this lecture, in this podcast, we're going to talk about innate immunity, and there's basically 5 take-home messages from this. The first is that innate immunity is a non-adaptable barrier to infectious agents. 2, there are anatomical, chemical, and physiological and cellular barriers that are used to protect us from infectious agents. 3, there are phagocytes, recognized pathogens. There are specific types of cells. There are particular receptors on their cells that are tuned to recognize molecular patterns on bacteria and infectious agents in order to get rid of them. 4, inflammation is activated by the interaction of these phagocytic cells with the pathogens. We will be discussing inflammation in more detail later on, but we'll get a little taste of it in this session. 5, inflammation is mediated by chemical mediators. So, one of the things that we really want to know, is who are we trying to defend against? And I'm sure you all have a good knowledge of medical microbiology, and you're going to be learning a lot about that in the next semester. But there are a number of different potential agents. Some of them, for example, are protozoa, protozoa, for example, the trypanosome, which causes sleeping sickness, and you can see here a picture of these fluffy little things are those. Let me just get a pen here. Those are these things here. Let me get the color here a bit here. Okay, these things here, and then these are red blood cells, which cause sleeping sickness. There's, of course, opportunistic fungal infections, such as pneumocystis carni, which causes lung infections. And there's also bacteria, such as Staph aureus, which you know about, and skin staph, which results in skin staph infections. These are all types of infectious agents. And so bacteria, viruses, fungi, protozoa, and mycoplasma and rickettsia, all of these are potential infectious agents that your body needs to be protected from, because they like to really feast on you. They view you as a source of food, as well as a source of place to reproduce. All the great things you need in life, right? Food and a place to reproduce. There are other things that are also involved they're infectious agents, and those are viruses like HIV, the influenza virus, things that exist in cells, for example, such as tuberculosis, mycobacterium, and things that can cause food poisoning, for example, like salmonella. So these are all, again, other things. And we're going to see as we go through that your body has multiple ways of protecting against these agents. So bacteria, viruses, fungi again. Now, these agents need to get into your body. They need to, in order to reproduce. And there are a number of ways that bacteria and viruses can be taken up by your body. And these are recalled to as routes of transmission. For example, the sinuses, the trachea, the GI tract, the respiratory tract through the lungs, the stomach, the esophagus, oral cavities, eyes, the GI tract, the GI tract, which is over here, the esophagus, intestines, oral cavity. There are a number of infectious agents that infect through that. There's respiratory tract, as I mentioned before, and also the urinary tract. And so these are all places in which your body is open to potential infectious agents or invasion. And so let's take a look at those. The gastric, the GI tract, gastrointestinal tract. The types of infectious agents that particularly like to go to be taken to be transmitted or be taken up by the GI tract are polio, hepatitis A, cholera, traveler's diarrhea. Really, anything that can be consumed. In fact, hepatitis A is probably more common than people might think. Polio is an oral fecal route and was mainly passed on by oral fecal route, particularly among young children and all. And cholera again also is also cholera is also an oral fecal oral fecal route. The respiratory tract is, for example, influenza or common cold, are particles which get nebulized and again you breathe in. So when the person who sits behind you or around you into your desk or walks by you and blows his or her nose, they release a lot of particles in the air which you can take in. In some cases in Asian, particularly in China, when I was in China, you'll see people walking around in masks. It's not necessarily to protect you from what they have, but to protect them from what you have, and ability to take in through the nose or the lungs. We don't seem to use masks as much as in other countries, but other countries do that. Urinary tract is also an important area. Syphilis, HIV, chlamydia, yeast infections are all passed on through the urinary tract. So another area is also blood, and those would be transfusions or insect vectors. Transfusions are not benign. Transfusions are only as benign as we know what's in them. And so, for example, HIV, some of the early HIV patients were infected because they got transfusions of blood that had HIV, that had been contaminated with HIV. There's no test. No one knew about it. So and that's true for hepatitis B, hepatitis C. We now have tests for these. We now know we can screen blood that's been infected that may have these agents in it. So and again, also insect vectors are also a good point. Mosquitoes. The major one is malaria, for example, or plague was fleas in terms of bites, and then transmission by that route as well. Now, the innate immune response is really important, and here's sort of a schematic. If you look at duration of an infection for normal patients, what you find is that the infection comes up and then it goes back down with time. Your body, if it doesn't kill you, it will be resolved, and you'll won't have the infection anymore, and your body will respond and get rid of it. Your immune response. If you lack an adaptive immune response, you can see here that the infection goes up, and then what you'll see is in most cases it kind of tails off. The infection proceeds, and maybe death or morbidity happens over the course of that in terms of the duration of the infection. And this is the number of microorganisms that are available, and you can see that there's a number of microorganisms that you could say, for example, in the blood or in the infection room, in the infection area. If you don't have an innate immune response, then the number of microorganisms just skyrockets. Okay, and so it's a very, very important response. And so it's the combination of the innate and adaptive immune response that's so important. So if we look at this from the standpoint of what happens during a general infection, and let's just take a point where we have someone, a cut or a break in the skin. In the first 0 to 4 hours of that, Here's some bacteria that's on the surface of the skin. There are cells that are immune cells that are in the tissues to protect you. There is a blood vessel. There is at some point a break, maybe a cut in the skin. Bacteria or infectious agent can get into the skin and interact with potentially the immune cells in the first 4 to 96 hours. This can lead to pus and response to the response by the immune response. The local infection, you can have then a local infection if it's not completely dealt with, that can occur within that, still go on within that that environment or that tissues. And greater than that, you can have over a course of a week or so, you can have then a response of an adaptive immune response, in which you have here, these are antibodies, which can go in and also further further attack the bacteria invasion. So there is some break that occurs. One of the two responses you can see here is one of the things that, as we're going to see, that protects you from these bacteria that are here on the surface is your skin, which is essentially part of the innate immune response, whereas your adaptive response are these cells, as we've talked about before. So what are the types of the innate immune response? There are several different types. Here we've got 5 different types that we're going to discuss: the anatomic barriers, physiological barriers, chemical barriers, cellular barriers, and phagocytic barriers. So we're going to discuss these in a little bit of detail. So let's first look at the anatomic barriers. These are part of your anatomy that of course, that's going to protect you. The first part is your skin. It's the most. It covers the most surface area in your body, and is the largest interface with the environment. With your environment, consists of several layers. It's actually rather complex. Consists of a dermis, which is the inner portion, and the epidermis, which is the outer portion. And it contains many layers that are topped by a pretty tough material called keratin, proteinous glycopeptide type material called keratin, which is really tough. What is necessary for the skin to really do its job? Well, it needs to be. Really, your skin is dead. The skin on your surface is dead. You want a dry exterior. This is really very important. It's really important because you don't want, if you have moisture, if you have high humidity, bacteria love to colonize that. For example, great places for bacteria to colonize your skin that are hidden places that are not dry, particularly, for example, your armpits, for example. One of the things that you produce, as we'll see, is a chemical called sebum, which is an oil, which has one of its, although it's bacteriostatic, one of its main issues is to keep the skin dry. And so if you have oil, it repels water, and so it keeps the skin dry. And so it's important for that. You don't want cracked skin, although during the winter here in Minnesota, that can be a real problem because of the low temperature, because of the low humidity and the coldness. The moisture can leave the skin and cause it to crack. So oils are very important. Another one are that there's defenses, as we'll see, other chemicals. Your skin that can come out, basically excrete from your skin, which is pH and antibodies and other enzymes, as we'll see. Now, this is just a picture of the skin, and you can see it's very, very complex. There is, the epidermis, which is this layer up here, and then the dermis, which goes through which includes this area here, and this is quite complex. Like here's hairs, blood vessels, places for nerve endings, and some fat, as well as places for oils to be excreted. So you don't want this part. If this gets broken, then you can have access potentially to the blood vessels in which a bacteria could enter if it wanted to. Another thing about these cells is that there's tight junctions between the cells, so that things can't get in. And so the cells, if we were to look at them, we would see that the cells themselves have a lot of, there's tightness between the cells, so that nothing can get between them. And that's very important, this tight junctions between these skin cells. Now, the other part of this is the basement membrane, which is under the epidermis. And this is a layer in the dermis. This is a layer of collagen, which is under the epidermal layer. Bacteria, this is sort of like a coat, a little bit of like that flexible coat of armor. And bacteria, I mean, this is something in which you really want to see. Excuse me. You want to have a good collagen, because bacteria, if they invade or puncture that the skin, they can create enzymes called collagenases, which will destroy the collagen, which is this a very tough protein, and be able to cause and enter into the skin. And you may have seen some of this if you see flesh-eating bacteria. It's not the bacteria that actually are eating, it's these enzymes that they excrete that actually pave the way or excrete or destroy the tissues as they go through, and one of those is this collagen. Bacteria induce inflammation to can penetrate the basement membrane as well, but these enzymes are really potent. Another area atomic barrier are mucous membranes. These cover the exterior parts of the body, which remain moist, and they're covered with mucus. For example, here are the nasal passages, here are the nasal mucosa, and they have these soluble glycoproteins and glycosamine glycans, which has adherent properties and provides basically false binding sites for viruses and bacteria, particularly viruses. And so these mucous membranes have cilia, which is flow of cilia, which allows things to be moved up and out of the area, out from the lung tissue, out of the area, out of the trachea, and allows for the bacteria, it allows for viruses to be moved out and eventually removed. But it's very important, these mucous membranes are very important because they allow things to come into you that are good, like food, air, but they also have to protect against bad things like bacteria and viruses that want to invade you. These false binding sites are really, in order to mount a successful infection, a key thing is the infection of viruses, bacteria. They have environments in which fluid flow must bind to some surfaces. So mucus and some other fluids contain glycoproteins that serve as false binding sites that can then carry them away. Viruses and bacteria like to live in environments with fluid and air as well. If there's air where there's fluid, particularly bacteria, that will allow them to mount this in defense. And so you can see here, if you have a pathogen, and here's a target cells for infected, that if this particular pathogen has a receptor in which it's going to bind to these cells and then get into this virus is going to bind to this receptor. For example, like a cold virus is going to bind and get into the cells and infect them. Then if we provide mucus provide false binding sites that basically comes in and blocks those and binds to them. It's sticky, it binds to the surfaces and allows them to and prevents them from going from binding to the, we want to prevent them from binding to the prevent them from binding and going into the cells and causing the infection. So now the next set of areas are called chemical barriers. First set, as we kind of mentioned a little bit, there's a little bit of overlap here between the mucous membranes and the chemical barriers. First are flow of fluids. This is a major factor in maintaining many environments pathogen-free. For example, and here's several examples here. The eyes, tears, your tears, a major problem for people who have dry eyes is infections in their eyes, and so tears, continuous tears, is important. In the vagina, there's fluids and cells of menstruation these fluids allow this to be removed from the vagina and allow the fluid to flow out of the vagina. In the urinary tract, there is urine. Urine is really important in terms of cleaning out the urinary tract. That's why that first part, if you go in to get a urine sample, they don't want you to take the first part, because when the urine comes through, it basically cleans out all the any bacteria or anything that's in the urinary tract. And so after that, urine is actually sterile. Skin, sweat is important. Fluid was one for it. It flows and protects the pores and allows fluid flows, as well as keeps you cool. In your mouth, you produce about 1 liter of saliva a day. So that would, if you just have it's about 30. That's about 32 ounces. So just think about that in terms of the amount of a large 2 drinks, large water bottle. That's how much saliva that you produce every day and is swallowed to remove things from your mouth. The respiratory tract, again, we talked about this as mucus and is propelled by cilia, then swallowed at low pH, at the low pH to the low pH of the stomach, which can get rid of it. So so all these areas have different types of fluid, usually in water and some mucus, that allow it to be flowed and to remove, to allow it to be pathogen free. So another area is low pH. Low pH is very toxic to some bacteria, and most grow very inefficiently at low pH, and they can't really stand it. So in your stomach, for example, if your stomach is around pH 2, when you have a meal, it drops to about pH 1, and so that's a good thing, because if there's any salmonella that comes in with you when you've just eaten, when you're eating your lunch, in your sandwich or your soup or whatever, this low pH functions to really provide a very hostile environment for bacteria, and can also lead to them being destroyed. In infants, this low pH can be caused by when you're fermenting lactose, and so this fermentation of the lactose by lactobacillus can also lower the GI tract pH and just provide not a very good environment for bacteria. And then there's glycogen fermentation in the vagina as bacteria or commensals, which also lowers the pH, and both protect against the colonization by pathogens. There are enzymes that are part of the chemical barriers. One of those, for example, are lysozyme. There are also RNases. Lysozyme, for example, is an extremely robust enzyme. It can survive in the GI tract. It can survive on your skin. It's a very robust enzyme, and it can hydrolyze. It hydrolyzes basically the peptoglycans that are important for maintaining the bacterial cell wall that are abundant on bacterial cell walls, and basically breaks up these cell walls by causing these sugars to be hydrolyzed. And what this causes is then the dissolution and destruction of the cell walls, which causes the destruction of the bacteria. And so they also work against some cases viruses have glycans that are on there for receptors. These can also work to destroy those as well. So there are also other enzymes like RNases, which will destroy viruses which contain RNase. Your hands are basically covered in RNase, actually RNase activity, and RNases are extremely stable, very stable proteins for that reason. Another chemical barrier we mentioned a little bit, we mentioned earlier, is sebum. This contains unsaturated fatty acids. It's part of the protective oily secretion, as we stated earlier, on skin. Sort of like you can see here. This is baby oil. It's just an example. It's not like it's like baby oil, but baby oil, causes to, makes the skin supple. It keeps it dry, and it keeps it from being cracked. And again, and it's bacteriostatic. Bacteria do not like to grow in areas in which there's it's oily areas. Another chemical barrier are the interferons. These are a type of protein or peptide that's released by virally infected cells to prevent virus proliferation in nearby cells. We'll take a look at that mechanism in a minute. And they activate. They prevent virus proliferation in nearby cells. They can cause the cells to basically. And that's why they're called interferons, because they try to interfere with virus or bacterial infection. They activate cells to inhibit protein synthesis and nucleic acid replication. And they increase the ability of cells to express immune receptors, and increase the phagocytic activity of phagocytic cells, like, as we'll see, natural killer cells and macrophages. So these interferons are really good because they really can help in modifying cells to do it. And this is kind of one of the first times you're going to see, and we'll keep seeing this, that there are proteins and small peptides that are really important in activating the immune cells, are an important part of the immune system. So here we see this in terms of the activation using interferon. You see there's a virus that infects. There is, when it infects, there's a series of activations that occur that lead then to the transcription of interferon alpha and beta. There's a paracrine and an autocrine component to this, in which the interferon beta leaves, binds to the receptor on the cell, and these then are, the uninfected cell is then, response is then given. It's basically a call for the cell to prepare itself, because there's an infectious agent, there's a virus nearby, and it goes through several different processes, things that can help it upregulate it in terms of upregulation of enzymes that will destroy viruses within the cell, parts of the immune potentially, for example, MHC1 types of responses, upregulation of proteins that are part of the adaptive immune response, so to protect the cell. Then there's an autocrine system that goes in that then further activates the cell to then produce interferon alpha, which has further effects in upregulating immune response of adaptive immune cells. So there are, the next part are referred to are cellular barriers. And cellular barriers are organisms, they're called commensals, and these are organisms that grow in and on the body and are harmless or even potentially helpful. They do three things. One, they prevent colonization by pathogenic strains. They basically take up the space in which pathogenic strains would want. So if you have a field, for example, and you have a lot of there's a lot of, I don't know, if you have a field and there's a lot of cows in that field and they're eating, you can't really put many more cows in that field. It's the same for bacteria. Bacteria from one type of bacteria covering an area, another type of bacteria can't really come in and grow on that area because there's no place for them. And so they prevent, and they also take up nutrients that could be useful. They also produce vitamins. For example, you have you don't produce. You have folate. You need folate. For example, bacteria produce folate that you take up naturally from them in the gut, and they also help There's also help in metabolism, particularly in the GI. This, of course, is in the GI tract. These things are provided in terms of helping to metabolize the foods that you take in. They're really really critical, in fact, in some cases. So the types of commensals found in the body depend on a number of environmental requirements, and these are 6 things that are listed here: oxygen, temperature, nutrients, and humidity and pH. All these things are really kind of important for commensals and their ability to grow. Do they have enough oxygen? Is the temperature right? Is it medium? Do they have nutrients, humidity, etc.? They're natural barriers to product these. When they take up this and use this, these become natural barriers to productive infections and prevent them from doing damage. So because they're involved, particularly with the nutrients and oxygen, they involved in taking up that space, and these are the things that are provided that can be parts of their environment that are necessary. And they also have some cases resistance to enzymes that allows them to flourish in those areas. For instance, the GI tract. You should know that, for example, the GI tract, the flora of the GI tract is quite enormous, and also the things that are on your body. And so, for example, in the GI tract, in fact, if we were to take you and grind you up and look for DNA. Look for DNA from you, from a person, we would actually probably find very little of your DNA, but actually most of the DNA we would find would be from the bacteria that inhabit you. Turns out it's a huge environment, and it's a huge number of microorganisms, and the environment in the flora, that particularly in the GI tract, is a very sophisticated environment with a lot of different, its whole ecology with many different types of bacteria. When you see, for example, if you get colonization, the number of commensals is reduced. So, for example, if we take an antibiotic, this is one of the reasons that the use of antibiotics can be a problem. For example, in this case, you have the commensals in the GI. In fact, take an antibiotic, it kills a number of really wipes out the population there. Then something like Clostridium difficile, it can gain a foothold, and when it does, it can start colonizing and producing colonies of itself, and that will then result in toxins that can then produce mucosal injury. That can start to destroy the cells. And when that occurs, then you have there's an adaptive immune response to that. But then what you have is bleeding, and then what you can see is some of these. You can get some of the bacteria actually leaking across this GI tract. But you can then also have blood coming out and damage, in which blood can then come out into the GI tract, and because the epithelial cells have been damaged. So these commensals are really important for really making sure that you are protected against these opportunistic infections. And here's a list of our little friends, the commensals. This is just a partial list, but you can see there's a lot of them. Some of them, like staph epidermidis, for example, inhabit almost all the different areas. Others are more specific and are only in, for example, the GI tract. There are also things that you should see. There's some of these you might notice, like Neisseria meningitis is also, you'll find that there, or staph pneumonia pyrogenes. And so these are things that are there, and so you're carrying these around, but because the other bacteria are around to prevent them from colonizing, you don't have an infection. It's one of the things that protects you against an infection. And so if you were to go in, you might see people actually carry a number of very infectious agents with them, but they cause them no harm, because you have your commensals as well as your other part of your immune response is intact. You will not need to memorize this graph. It's just there for your to show you examples. Now, here's a series of review questions, so we can stop at this point. These are 3 review questions, and what I urge you to do is to answer these questions to the best of your ability. In the next slide are the answers to the questions. So you might want to take a moment here and stop, answer the questions, maybe go back and review a bit of the material to answer them, and then go forward. So you might want to stop now to do that. From this point on, I'm going to go forward, and we'll go to the next part of the innate immune response. So one of the other areas, as I mentioned before, are the phagocytic barriers. Phagocytic barriers are cells which carry out phagocytosis. And there are basically 2 types of leukocytes in the blood. 2 types if we have. One of those are the neutrophils. This is a type of granulocyte. We're going to see more of this later. This is a really important set because they're very important. They're very important cells in your fight against bacteria. And then monocytes, which are also a type of agranulocyte, and these will become monocytes are in the blood, and they're also phagocytic cells, and they later can differentiate into macrophages. So phagocytes then can be divided into sort of 2 different types of them. These are macrophages. These are phagocytic cells like neutrophils that are primarily against bacteria in the early phases of an infection. That's why it's important to have a good number of those neutrophils. You probably to help fight against the infection, and you can see why now. For example, you might have had someone in your family, someone you know, who's been under chemotherapy. Chemotherapy, in a lot of cases, is quite toxic to immune cells like neutrophils, and if the neutrophils drop to a very low level, then you'll be much more prone to infection, and that's why there are those need to be monitored, and there are even certain types of growth factors that can be used to build those up, as we'll see later. Macrophages are the other one. These hibernate or plow or go in and out of the tissues. They're fixed in tissues. They're referred to as reticular endothelial system. The liver and spleen are full of them, particularly as well as the tissues. Kupffer cells are another type. Kupffer cells are another type of cell that can do this, and which primarily resides in the liver. They're also active against microbes, dead and dying macrophages, and dead tissue. They kind of also are a garbage collector. They eat up, things that dead cells, take them in, metabolize them so that they're the parts, the metabolites can be used again. The functions of these phagocytes. One, as I mentioned, they're to clear the body of debris. Because there's a lot of junk that as your cells get to die and get turned over, as well as bacterial junk that needs to be destroyed. 2, they secrete defense proteins like interferon. We've just seen that allow us that will protect cells. 3, they ingest and kill bacteria, and so we're going to see that, primarily that. We're going to look at that mechanism. They are also a part of inflammation, because they synthesize endogenous pyrogens like IL-1 that go off and result in other cells being activated. And as we're going to see later, particularly for macrophages and others, they also do something called antigen processing. We're going to come back to this, but this is one way, just to give you sort of a little taste of what's going to happen in, when we start talking about the adaptive immune response, where they interact, or there enters an intersection with the adaptive response. So what happens first? So if you have a bacterium or a particulate pattern, bacterium or particle of matter, it will bind to a receptor, and we'll see a bit about what kinds of receptors in a minute. But these phagocytes can retain receptors that will bind, will recognize certain components on bacteria and viruses. They will then endocytose them. They will go into the cell, forming an endosome. That endosome then that triggers fusion with a structure called the lysosome, which contains a lot of noxious enzymes, particularly enzymes that are able to degrade cell walls. It also contains the ability to produce enzymes that will produce noxious chemicals that are very noxious to the bacteria. And so these fuse, forming the primary lysosome. This bathes basically the bacterium in a sea of noxious stuff, and then that leads to hydrolysis and destruction of the bacteria, forming what's referred to as the secondary lysosome. Those are then moved to the surface of the phagocyte, and then excreted out of the phagocyte. And these kind of there may be nutrients, little pieces of DNA, protein, amino acids, peptides. There are then sugars that are excreted. They can be then taken up and catabolized or removed further. So here, for example, here is a macrophage, this big red thing. Now the macrophages aren't red. This is false coloring, but it gives you a point. You see they have a very fluffy membrane, which has lots of indentations. So their actually membrane surface area is extremely high, and they contain lots of different potential receptors. And you can see here's E. coli. It's moving. They move quite rapidly. They're running into E. coli. They're then taking up these E. coli. You can sort of see one that's being sort of taken up here, one that's in the surface. They're binding to the surface, and so it's going to bind and take up these. So they're quite large compared to a. So you can see here a little close up. You can see all these little indentations from the membrane. How tremendous the surface area is, and you can see the bacteria being engulfed by which are in green here, being engulfed by the macrophage surface. So this is a little. Well, this isn't working quite right. I'll show this in class, but this is a macrophage. It worked before. I don't know why it's not working now, but that's okay. I'll show this in class. But this is a macrophage that will be eating or ingesting part of a yeast. This is an example. Now, during phagocytosis, as I said, there's not only enzymes that are produced, but there's also chemicals. And one of those is there's toxic oxygen species that are produced. One of the things that is produced, one of the enzymes that's critical is this NADPH oxidase, which takes oxygen and reduces it to superoxide. And so the superoxide itself is not tremendously toxic, but in the presence of, for example, superoxide dismutase, it then is converted into hydrogen peroxide, and hydrogen peroxide and superoxide can combine and forming a very toxic species called the hydroxyl radical. This is a radical, and singlet oxygen. Both of these will react very rapidly with proteins, glycans, DNA, and so these are all species. These oxygen species are all very important. Superoxide, anion, hydrogen peroxide, singlet oxygen, and
the hydroxyl radical. So these serves as chemical. These serves as chemicals that are that will go after and destroy things. For example, the hydroxyl radical, as soon as it sees a proton, it grabs it, rips it off, and so it can be quite toxic. Now, this is referred to as the oxygen-dependent myeloperoxide-independent reactions. So these are independent. Myeloperoxidase, we'll see in a second, but these are dependent, and they're independent reactions. There's no myeloperoxidase here. The dependent reactions, in which myeloperoxide takes hydrogen peroxide, as you can see here, and combines it with a chloride and produces hypochlorous acid, which is this in red here. And this is really more or less bleach. This is essentially, you use bleach to whiten things and destroy things. This is essentially an acid that's very potent. It's very noxious and will destroy proteins, glycans, and DNA. In addition, when it combines with water, it produces singlet oxygen, which is also very toxic. And so you can get this whole menagerie of hypochlorous acid, singlet oxygen, hydroxyl radical that are produced that are quite toxic. And this is produced by the macrophages, particularly in the lysosomes. So these chemicals. Now you do have ability to detoxify. Why doesn't the macrophage just destroy itself? Well, in the cytoplasm, you do have superoxide dismutase serves the function of actually detoxifying hydrogen superoxide by producing hydrogen peroxide, and this hydrogen peroxide is then removed by catalase. Both of these reactions are extremely fast reactions, and hydrogen peroxide in the cytoplasm is removed, and so this will protect the macrophage against its destruction of its own proteins and lipids, particularly its own proteins and lipids and membranes, etc. Now, there are another toxic species are nitric oxide. Nitric oxide is produced by nitric oxide synthetase. This is relatively new, I guess. Not for you, but for me, because I remember when there used to be no nitric oxide. But in the 1990s, it was discovered that nitric oxide, there was an enzyme called nitric oxide synthetase that produces nitric oxide, which is also a radical, and it's toxic into infected cells in the vicinity of macrophages. It's produced, it reacts very quickly, and so it doesn't diffuse very far. But it's induced by these macrophages, and it can be induced. You see here, this is interferon gamma, which can cause the induction of this, as well as this tumor necrosis factor, which can also TNF, which can also cause this. So it's an inducible one. So these interferons can cause cells to induce their ability to produce nitric oxide synthetase in these chemicals. Now, macrophages express several receptors specific for bacterial constituents on their surface, and this allows them to recognize pieces as well as pieces of bacteria, as well as the bacteria themselves, as well as parts of DNA. And here's a sort of a list here at the top. There are receptors that will bind to lipopolysaccharide. There are receptors that will bind to, they're called scavenger receptors that will just bind things that are polar, particularly things that are positively charged, for example. There's glycans that will bind sugars. Here's one that binds mannose, which is on a lot of in bacteria you'll find mannose, and so it will bind that. And then there are things called toll receptors or toll-like receptors. So these will all bind. They can bind. They're part of things that will bind to bacteria and bring them into the cell, forming the forming the lysosome, engulfing the cell into the endosome, to the lysosome. This recognition is referred to as pattern recognition, and these are referred to as pattern recognition receptors, or PRRs. These receptors are with specificity for they have specificity for pathogenic molecules, referred to as pathogen-associated molecular patterns. So the molecules, okay, on the surface, the pathogenic molecules, the molecules that are on the surface of the pathogen are referred to as pathogen-associated molecular patterns. So what those patterns are, so PAMs. The receptors are pattern recognition receptors, okay? So don't confuse those. One is the actual type of macromolecular structure on the bacteria that's recognized. The other is the actual receptor. And these are present on the cells, and binding the pathogens results in an immediate response. So these are on all these all macrophages contain these, have these or express these. One of those types, which is really significant, in terms of the number of them, are referred to as toll-like receptors. Mainly, they're called toll-like receptors because they have similarities to a receptor that's found in Drosophila, which are fruit flies, what are called toll receptors. And so these are referred to as toll-like. They're similar too when they first were discovered, but they don't have anything to do with fruit flies, so don't worry about that. So toll-like receptors, they have a specificity have a specific specificity for pathogenic molecules on pathogens. So here they have this kind of interesting crescent shape, and here you'll see that there are several types of these. There's toll receptor 1 and 2. So there's sort of two types. One in which you form, the toll-like receptor is formed from two different types of, or the recognition first needs two different types of receptors. So for example, if we looked at diacyl lipopeptides, they come together, TLR and TL2 combine to form the receptor that you need. For toll-like receptor 7, for example, this one that's inside, it's two, these are homodimers, so they you have two TLR7s that are necessary. It'll become a little, I'll show you in a second what that's meant by that. But you can see here there's several different ones that will bind to parts of bacteria, flagellin, lipopolysaccharide. And then there are ones, upon engulfing, that are actually, so these are on the surface, so these are on the cell membrane. So this is on the cell membrane here of the back of the macrophage. These are in the endosome. So in the endosome, they're also, these toll-like receptors are expressed in the endosome. So for example, when a virus is taken up and destroyed, then its DNA or its RNA becomes unwrapped and potentially accessible. And so then you'll see that you have receptors that will recognize, basically recognize nucleic acids, because they aren't on this. You're not going to have nucleic acids on the recognition on the surface of a macrophage because it's in the bacteria or the virus. But you do have it once it's in the endosomes and then it's just being potentially coming apart, then it becomes available. And so you can see here the ones that recognize nucleic acids are in the endosome, expressed in the endosome. So some are expressed on the surface of the cell, and some are expressed in the endosomes. Now here's a bacterial lipopolypeptide. So remember, I said that some of them, let's see here. For example, in this case, this is bacterial lipopolypeptide, which can be recognized. So this is, so in this case, this is a crystal structure. And what we see in this crystal structure is we can see what we see is the we see TLR1 and TLR2. So TLR1 is in red, TLR2 is in blue, and they've come to form a binding pocket. They've combined to form a binding pocket that will, as you see here in the middle, is where the lipopolypeptide is bound. And so they form this it's sort of two crescent shapes that come to form this binding site for the small molecule from the bacteria. In this case, this is TLR3, in which case the TLR3 this is a homodimer, so the one that binds the bacterial lipopolypeptide is a heterodimer containing two different TLRs. Important. If we look at the TLR3, it binds a double-stranded RNA, and here you can see this right here. It binds it across here, binds it across the peptide protein, and it's actually two of these crescent molecules which are the same. They're the same molecule, so it's a homodimer are involved. In either case, you can see that they can bind small molecules, and the toll-like receptors are engineered so that they can bind large molecules like nucleic acids, pieces of nucleic acids. This is a little chart that shows a number of them, what the ligands that they bind, the microorganisms that are recognized. So TLR3 was a homodimer we just looked at. It binds to viral RNAs, double-stranded RNAs. Let's see, TLR2, TLR1 are heterodimers. We just saw TLR1, and what we find here is that this is peptoglycan, and that it binds from the gram-positive and gram-positive bacteria, yeast, or trypanosoma. I want to point out that some of these, you notice here, the intestinal endothelium, macrophages do produce these. We've looked at this for macrophages, phagocytosis. But there are a number of other cells that also to produce and recognize, produce toll-like receptors as well, not just macrophages. We're not going to get into that, but just be aware of that. Now, the activation, if you could what do they do? These toll-like receptors, when they're activated, activate the immune response as well. Their main function is to activate the macrophages to become more activated, to produce interferon, to become just more mobile and more ability to produce lysosomes, to activate the amount of lysosomes that they produce, etc. And so these have recently become, in the last 20 years, potential referred to as immunomodifiers. Drugs that can activate them are referred to as immunomodifiers. And we can see that here. This is imiquimod, or Aldera, which was originally developed by 3M Pharmaceuticals. And this is approved, and you notice its main function is it recognizes it's similar to, maybe it looks a little bit like a nucleic acid. Look at this point here where where you have a purine type of substructure, and this is a useful in for papillomavirus and infections, referred to as genital warts. It activates alpha interferon, other cytokine released by macrophages. So it has a very good antiviral and anti-tumor activity. It's only for external use, or applying to the wart, or applying to, for example, in some cases it's used with melanoma. It's been tested for melanoma activity, but you don't want to ingest that, because you'll get activation of the entire immune response systemically, and you don't want that. You only want it locally. I had a former student of mine working out there on this, and these things can be really, really, really activated. He made some other analogs of this molecule, and when he just, at one point he had made one, and he forgot to clean his hands and touched his lip, and his whole lip side of his face blew up, because it activated his the macrophages, which activated cytokine release, which activated an inflammatory response. So you don't want things that active, but there are a number of these now that have been used, and this just gives you a taste of what what these kinds of things do. They activate toll-like receptors, and in fact, Aldera works by binding one of those toll-like receptors, toll-like receptor 7. So it functions sort of like as an RNA mimic. Now, you won't have to know this in any detail, but just be aware that when you get these toll-like receptor activation, that there is a cascade of signal transduction events that occur that lead to the transcription of inflammatory cytokines and the production of those cytokines that will then further activate cells, activate immune cells, as well as produce molecules that will tell potentially infected cells to protect themselves, like the interferons. So this is their main function, is to just activate this immune response, which leads to then inflammatory cytokine response, which then leads to activation of other cells to come in and destroy what's going on in the infection. There are some of these cytokines that are produced. There are a number of these, and these, just if you don't know what that is, a cytokine or chemokine are peptides or small proteins that bind to cell receptors, causing a variety of biological responses. And the ones that macrophages produce are, for example, IL-1, which activates the vascular endothelium, activates lymphocytes. It's involved in fever production and production of another very important inflammatory molecule, IL-6, and local tissue destruction. TNF-alpha activates vascular endothelial cells, increases the permeation, so that cells can get in, allowing other molecules like antibodies to be able to get into the infected area. It also has an effect on fever and shock. IL-6 is a sort of a lymphocyte global activator and increases antibody production, and it can induce some. If it's not controlled, can actually induce some rather severe acute inflammatory responses, and as well as fever. There are also chemotactic factors like CXCL8, which recruits neutrophils into the response and other immune cells, and then things like IL-12, which activates these other cells like natural killer cells and other immune cells. So it's sort of you get a whole gambit of stuff that's important here for activate that macrophages can contribute to the inflammatory response. Excuse me. So in these physiological barriers, inflammation is the primary response to the damage of tissues by microbial infections and physical agents or chemicals. Symptoms, of course, are redness, pain, heat, and swelling in those areas. Really what you want to do is, the whole thing is to contain the bacterial infection and the infection in that area. So that's one reason you have swelling and heat, to provide an increase in heat. The fever provides bacteria don't like to, for example, don't like heat, or virus infections, particularly viral infections. They don't efficiently assemble in tissues that are infected, and the swelling is to contain that, to try to contain it and wall it off from the rest of the body. Fever, for example, as I said, is controlled by hypothalamus. Elevated response in temperature in response to exotoxins like bacterial infections or leukocyte pyrogens, like I said, IL-1, for example. It occurs during inflammation, and this higher temperature, as I said, inhibits proliferation of viruses, but it also speeds up the bodily functions and the immune response, causing greater metabolic activity. So this temperature is important. Actually, the temperature is important in that area. The temperature is important, and it's not just a bad thing. It actually contributes to resolving the infection. So again, functions of inflammation to destroy and remove injurious agent, confine what can't be destroyed, and repair the damaged tissue. I didn't mention this. So swelling, etc., and the heat are to destroy and confine and get rid of it. But there's also a process. There are cytokines that are produced in response to repair that damaged tissue. Is the other part of this, not just to get rid of it, but then to repair the tissue and bring it back to normal. So there are a number of mechanisms by which bacteria have evolved to get around phagocytosis and thus stop inflammation from occurring. Some of those ways are to kill the phagocyte. They can produce, once they're inside the bacteria, they can produce a toxin that will kill the macrophage. In some cases, overwhelm, as like 3. If there's too many bacteria that are taken up, or, for example, what can occur is the bacteria can reproduce rapidly within those endosomes, and when they do that, they can, or in the cell, they can kill the phagocyte. They can inhibit binding, for example, or endocytosis. So, for example, some bacteria will produce enzymes that will destroy those, that will hydrolyze the receptors that recognize them, so that they won't be able to bind to the macrophages, as an example. And in addition, they can contain, their cell walls have evolved, in some cases, to be resistant to digestion. For example, TB and leprosy, leishmania, what you find is that those cell walls are really highly resistant to being destroyed by macrophages. And in fact, for example, for TB and leprosy, what you find is that they are they actually live in the macrophages. They find a home there and reproduce there, and the macrophage kind of goes haywire in trying to get rid of it, but it can't get rid of it. So So within this part, here's 3 review questions, just to allow you to go back and look at what we've talked about. Gives you an idea about to go back. So go ahead. You may want to stop right here and see if you can answer these review questions. There's 3 just short ones. The next slide will have the answers to the review questions. I'm going to proceed now on to just sum up at the end. So as we've seen in the innate immune response, the take-home message, what have we seen? We've seen 1, just to summarize, the innate immunity is a non-adaptable barrier to infectious agents. There's nothing that what we've seen there happens either as a person develops and is with them with their whole life. 2, there are a series of different types of these categories: anatomical, chemical, physiological, and cellular barriers. Phagocytes are important because they recognize pathogens by these receptors that are tuned for molecular pattern recognition. There are noxious chemicals like reactive oxygen and nitric oxide that are used to destroy and ingest pathogens. And inflammation is mediated by these chemical mediators and is important to controlling, destroying, and resolving the infection, as well as for repair of the tissue. So with that, we're going to end. And I will see you in class. In class in which we'll do some activities that will cover this area in different aspects in a little bit greater depth.